Well, my name is Sarah McKim, and we're in a lab in the Division of Plant Sciences at the University of Dundee, which is situated in Invergowrie at the James Hutton Institute. I work on barley, so my research group is interested in understanding how different genes control architecture in barley, so control how barley looks, essentially, and how it develops over time. When I first arrived in Dundee, I had no experience with barley at all. And just decided to jump in and just start growing it and seeing what it looked like. And I remember vividly the first time I was using a scanning electron microscope to look at the developing flowers in barley. So barley produces this, what we call a spike, so it's that grain head that you can see waving in the fields. That starts off extremely small, right, millimeters, less than millimeters. And so you have to use a microscope to kind of see the detail that's going on. And that structure was the most bizarre thing I've ever seen underneath the microscope. And I just, from that moment on, wanted to understand how that was happening, how that was forming. It's an exquisite piece of work, I think, of reproductive architecture. It's fascinating. Just in terms of when you discuss a building's architecture, you're interested in how the different parts are put together. The relationship of the parts to the whole is how I would describe plant architecture as well. In the case of organisms, they have to grow like a building, putting their bits together, but they have to do that while alive. So they have to be functional throughout the building process. And so it's, to me, I've always been fascinated by development, the idea that you can start with a single cell and end up by a variety of mechanisms generating a coherent multicellular organism with specialized parts that function together in order for the organism to reproduce. Part of the reason that I am working on barley now, I used to work in Arabidopsis, which is your model plant, was that I was interested in working in a field that was kind of more transitioning towards application. So working on traits, various aspects of plant development that could be useful. But my main interest is a curiosity, a desire to understand how the organism develops. My lab is a relatively new lab, so we've been up and running maybe almost 4 years now. So, and the type of work that we do isn't at the breeding end. We're interested in, we use mutants, we use different cultivars, different types of barley to, as a way to kind of compare how they develop differently, and then we pair that up with the genetic differences. So we're able to kind of put a function to a gene's role during development. So we're interested in kind of understanding the molecular mechanisms going on as barley develops. Now we can then use that information to kind of highlight new genes, maybe better genes to achieve the same sort of trait modification, rather than kind of relying on finding a gene variant by chance. You can go using the knowledge that we've accumulated about genes and protein function and how to alter that. You can go in and do the exact change that you're curious about and then see the output in the living organism. What does it look like when you change that gene in a very specific way? So it's targeted, and so that gives us a lot of predictive power, or it lets us see if our predictions are correct or not. As the transformation technology comes along, you can do that in the crop, in the actual line of barley that might be grown for food purposes at the moment.